We'll be starting in a few minutes. Good evening and welcome to the third of our series of talks about UC Santa Cruz's response to the pandemic. We've previously heard from Rebecca Dubois, whose lab is developing antibody tests, and Marm Kilpatrick, an epidemiologist, who gave us a sobering look at our possible exit strategies. Tonight, we'll hear from a group of scientists who came together in the early stages of the virus to upright a diagnostic testing operation. This was This is and was an example of science unfolding in real time at UC Santa Cruz and leadership in the community to address a shared concern. I'm excited to share this work with you tonight. I've had the pleasure of working with Isabel Bork in her role in Genomics Institute. Tonight she'll tell us a little bit more about our speakers, but I wanted to talk about one in particular before Isabel does her presentation. Jeremy Sanford's a professor of molecular, cell, and developmental biology at school, but I first met him at a rugby reunion of all places. I played ball 50 years ago, and Jeremy's the volunteer coach. And what struck me was how this tradition of volunteer coaches by very distinguished scientists had continued in Santa Cruz from the time I had been there. My two coaches were both well-known and well-regarded physics professors, Ron Ruby and Dave Dorfan. And today, Jeremy carries on that tradition. But meeting him in that context made me interested in his academic work. And I actually later on visited one of his labs that he had opened up to undergraduate students, which is not an opportunity, a research lab, which is not an opportunity most undergraduate students have. I was so impressed by the educational opportunities he was presenting and the work that was being done there. But I was also struck by the fact that Clark Kerr and Dean McHenry, the founders of Santa Cruz, would have been so happy to see this, because in Kerr's mind, the most important thing at the university was to advance human knowledge and to educate the children of California and the world. And both of these things were being done in one context. So in that regard, I can't, my connections, it made my connections to the university that much more poignant to me, and it made me that much more prouder of the work that's being done here. But now I'd like to turn it over to Isabel to say a little bit more about our speakers as they share perspectives on the pandemic and their pop-up lab. We'll have plenty of time for questions at the end. Isabel?
Thank you all.
Thank you, George, and thank you, Rafe and George, for sponsoring this lecture and for that introduction. I'm not surprised that you know Jeremy through rugby, of course. Before we get started, I just wanted to share a couple details and make sure everybody knows that this is being recorded. And Also, often there's a chat function in the Zoom presentations. There isn't tonight because we're using a webinar tool. However, there is a Q&A section, and we really encourage you to send questions to us in the Q&A. Our plan is to split this talk tonight between a literal talk and then add 45 minutes or so for questions, because we really want to be able to answer what's on your mind about COVID-19 testing. We know it's at the top of everybody's minds right now, and we really want to spend some time tonight contributing to your specific interests. So in terms of starting us off, my plan is to give you an introduction, and then this is a strange kind of Zoom presentation for a Kraw lecture because there are 4 of us talking today. There are 4 of us talking today because we are talking about the pop-up lab that we created for COVID-19 on the UCSC campus. And the 4 of us did this together. So we wanted to present to you together. So first of all, you've heard about Jeremy already. You've heard about rugby, so you know that's key to Jeremy's being. He's a professor of molecular cell and developmental biology, and he is very much our vision person on this lab and has fought really hard to bring this to the campus. We have Elena Vasky, who's an assistant professor of molecular cell and developmental biology, and Elena fills an incredibly important role here because she is our CLS, which you will hear about it later in this presentation. And without that, there is no way we could have done this. And Michael Stone is a professor of chemistry and biochemistry, and he has been absolutely instrumental in making all the technical worlds that we live in here actually work. And you will hear more about that as well. Next slide. So we want to tell you a little bit overall about our mission. We are passionate about serving the Santa Cruz community and increasing testing capacity. We are also very concerned about giving a quick turnaround of results. I know everybody here has read that there's nationally and locally not enough testing and that returns of tests are taking a long time. That was a problem we were committed to solving, and this campus was committed to solving. We also wanted to serve not only the student and the employee and the university community, but also populations vital to the recovery of Santa Cruz. Next slide. So I'm not going to go through everybody's name here, but the reason this slide is here is really to say that this was such a group effort. This is such a group effort. And so what we have here are people who have not only started the Molecular Diagnostic Lab on campus, but people who have made this all possible. So that's from the people I just mentioned, the 4 of us, but we've also gotten input from legal counsel has been amazing on campus. The IT group has been amazing on campus. We have a lab technical team and an operations team made up of people who have nothing to do with COVID-19 testing, obviously, who helped kick in from all sorts of areas of work and have really stepped up and worked around the clock. And so we thank all of them. The UCSC administration has been extraordinary and has acted very quickly and with passion to serve the people of the campus, but also the people of the community. And we thank them deeply for their partnership in all of this. I mentioned earlier that we have partnered with the community because part of what we care about is serving underserved populations and making sure that we are filling the gap that is necessary in testing. And so we want to do that not only on our campus, but in the Santa Cruz community. And so I wanted to say just a little bit about our community partners. We have Santa Cruz Public Health, which is Santa Cruz Community Health. And that was the first partner that we had. And that is a local clinic that actually began from students at UC Santa Cruz and was started by women that were students at UC Santa Cruz, and now serves 12,000 people in Santa Cruz without regard to income or race or sexual orientation or gender identity. So it's a full service clinic. In addition, we work with Salud para la Gente. And so where Santa Cruz Community Health serves 12,000 people, Salud serves 28,000. They also serve an underserved population. They are available to everybody, but they do have a focus on the ag community, and they have a large farm worker and immigrant population. In addition to that, we have worked closely with Santa Cruz Public Health, which is the county, and we are collaborating deeply with them, and we are grateful to them for all the work that they are doing to protect Santa Cruz. And then finally, Santa Cruz Community Foundation has just been incredible in terms of supporting the COVID-19 work in Santa Cruz. And they provide funding and bring groups together to support community initiatives, and we thank them deeply for their support here as well. So with that, we would like to get towards telling you what our Molecular Diagnostic Lab actually does. Thank you.
All right, thanks, Isabel. Thank you, George. It's great to be here with everyone, even if it is a bit remote. What I thought I would start off with trying to provide a little bit of the backdrop or the backstory for how Molecular Diagnostic Lab sort of sprung up in the heart of our sort of beautiful research campus. And I think the backstory sort of begins with a group of RNA molecular biologists that met. We meet once a month for exchanging ideas, and one of our colleagues gave a presentation about the data that was emerging from Wuhan, China. And he pointed out, this is in January or maybe late December, that maybe as a group we should begin to think about what kinds of actions we could potentially take. And I think at that time, for most of us, we were busy with grades and teaching and things like that, that we really kind of put it out of our mind and hoped that it would go away. It was sort of mid-March when the lockdown was announced and all of our lives really changed drastically, that we really finally thought, well, we definitely are going to need to take some kind of action here. And one of the most obvious problems that we'd heard about, obviously through the media and also anecdotally through our peers, was that not only nationwide but in Santa Cruz there was clearly problems with having sufficient testing and even having a turnaround time that was sufficient to act on. We'd already heard stories in March about diagnostic labs like Quest being able to sort of people waiting eight or 9 days for those test results. And so Santa Cruz has some deep expertise in the area of nucleic acid chemistry and molecular biology. And so our goal was to take this expertise and create a nucleic acid testing lab that could really help our community. Obviously, the campus had been it had been sort of locked down as well, coming out of the winter quarter into the spring quarter. And I think we all recognized that the campus is really a shell when it's without its students. It's a shell of its real self. And so a big emphasis on this was also to think about how we could potentially create an environment and a facility that could bring students back in a safe way to give us our sort of full campus that we know and love. And finally, the other bit was that we saw a real opportunity for forming a partnership that involved both our local government, academia, and in some cases even the private sector. So it's really been a chance to get that trifecta of collaboration. All right, can we go to the next slide, Michael? So just a little backdrop on the SARS-CoV-2 virus. Okay, so I think almost everyone here may recognize this type of slide. This is showing the central dogma of molecular biology, and I think most of us recognize that the instruction manuals for our cells and for our biochemistry is stored in our genome, which is encoded in DNA. And that in order to release this information and to manifest it, the information has to be copied into RNA, which is then often translated into protein. And those proteins are what do the job. The interesting thing about the SARS-CoV-2 virus is that it's a beta coronavirus, and its genome is actually RNA. It doesn't have a DNA intermediate. It is exclusively an RNA virus. And this presents some opportunities that I'd like to, in the next slide, just to mention. So we were encouraged to start this project because the research environment at UC Santa Cruz is really second to none when it comes to RNA molecular biology and to genomics. So often we get the question, how are you going to cure a disease, or how are you going to set up a diagnostic lab? You don't have a medical school. There's no big clinical laboratory in place or anything like that. But what we do have is a truly outstanding group of RNA molecular biologists that are part of the UC Santa Cruz Center for RNA Molecular Biology, as well as deep expertise in genomics, which comes from our Genomics Institute. And I think this group of very active and driven scientists is what gives us the motivation and the expertise to be able to act on this problem. So, what I think, the next sort of, I'll come back to you guys in a little bit, but I'm going to hand this over to Michael Stone, who is going to tell you a little bit more about the molecular diagnostic tests that have been developed in the lab, and probably provide a little bit of understanding about what it is that we're really looking at in these tests.
Great. So thank you, Jeremy, and to George for this opportunity to present. So yeah, as Jeremy mentioned, what I'm going to do with my short section of the presentation tonight is to lay a basic foundation. I'm sure many of you that are listening have been learning quite a lot through the media about diagnostic testing, and that there are many forms of testing for the SARS-CoV-2 virus, and some are reported to be better than others. So the first thing that I thought I would do is I would just sort of lay out with this basic graphic. Just going to grab a pointer here, which is essentially a generic cartoon schematic section of a coronavirus, which SARS-CoV-2 belongs to, that family of viruses. And so there are some of these features of the virus that I'm sure many of you have become familiar with in the literature and the media. And so the important points to note are that exactly as Jeremy just pointed out, the genome and the genetic information is in the form of an RNA molecule that is housed within this viral capsid or virion. And that capsid consists of a number of different types of molecules, which are membrane type lipid molecules together with a number of different virus-specific proteins, like the nucleocapsid protein, or maybe you've heard of the spike protein, for example, because a lot of the vaccine work that's going on is looking at ways to raise immunity against things like the spike protein. And so when we think about molecular diagnostic tests for COVID, or the presence of SARS-CoV-2, what I've done here is essentially break it down into three major categories. There are tests that directly ask the question, is there the viral RNA genome? RNA is a nucleic acid, so we call these nucleic acid tests. And as I said, this is a direct test for the presence of the viral RNA genome. There are many ways to do this, some more sort of novel and sort of newly reported. And a lot of the reports that come along with some of the new technology that is coming out is addressing the problem that Jeremy mentioned earlier about access to testing, turnaround time, issues with supply chain and things like that. And we're happy to address this in more detail in the question and answer session. But I just point out here this particular technique, which is called real-time quantitative polymerase chain reaction, or RT-qPCR. And I just make the note that this is still very clearly the gold standard of RNA testing, and I'll explain that to you in the next few slides. But there are other ways of testing. There are so-called antigen tests, and what that form of test is doing is essentially also directly testing for the presence of the virus, but in this case, typically testing for the presence of the viral protein, such as this nuclear capsid or spike protein. And then there's an entirely different category of tests, which you may have already listened to through our colleague Rebecca Dubois, together with Professor Jacqueline Kimmy and Martin Kilpatrick, talking about so-called serology or antibody tests. And what this test is doing, as you may be aware, is not actually testing for the presence of the viral in that moment, but asking the question, was the virus present such that an immune response has been mounted, and can we detect antibodies? And there's a lot of very interesting science behind serology and serology tests more generally, but today we're going to be focusing on direct SARS-CoV-2 detection. So really the essence of the power of this gold standard method for direct RNA detection of the SARS-CoV-2 virus is a very long established workhorse technique in all molecular biology labs called the polymerase chain reaction, or PCR. And really the power of PCR boils down to the notion that there's an exponential amplification of a DNA or an RNA molecule that essentially can start from just a single molecule here. And then in the course of about an hour, through a series of doublings, so that's why we think of this as exponential growth, where 1 goes to 2, 2 goes to 4, so on and so forth. If you do the math and you go through just 40 cycles, which in our typical test takes about 55 minutes in the lab, you go from 1 molecule all the way up to 1,000 billion or 1 trillion molecules. And as you might imagine, the ability to then subsequently detect the presence of that trillion molecules as opposed to the initial 1 molecule is vastly easier in instrumentation. And so really at the heart of these gold standard methods that are based on the polymerase chain reaction is this notion of exponential amplification of the viral genome. So here what I have is a cartoon schematic that I'll just walk you through, which is our current workflow for this so-called RT-qPCR method that I've just described. And so as many of you, I'm sure, are aware, COVID test starts with sample collection. Most typically, this is still a so-called nasal pharyngeal swabbing, where a flocked cotton swab is put deep into the nasal passage to collect sample. You may be aware that there are a number of other specimen types that have been approved by the FDA, and this is something that we can come to maybe later in the talk. But either way, you start with some patient sample, and then you go through a process of essentially breaking up all of that human sample and purifying out only the RNA. And I'll just point out here that when you do that, you're purifying hopefully no viral RNA in the best case if there's no infection, but if there is an infection, you're purifying the viral RNA together with all of the RNA in our own human cells. So you have a mixture there. And then that RNA molecule is then so-called converted or reverse transcribed into a DNA molecule. That's this process of reverse transcription. And now you're ready to do that PCR reaction that I just described, where that 1 molecule or a few molecules can be exponentially amplified. And then the last bit that I'd just like to point out is that the way that we detect, which is a very common assay called the TACMAN PCR assay, it's a variation on PCR. TACMAN is sort of a play on the old video game Pac-Man, where in the course of this assay, as the DNA is being copied and amplified, there's a probe molecule that all you really need to appreciate here is that that probe molecule gets degraded by this little Pac-Man enzyme, and the degrading process or the degradation of that probe releases a dye molecule and it lights up. And so the machines that we use to study this type of process are directly detecting fluorescence or the presence of light. And so here at the bottom of the slide, when you've done this amplification process, you either get a positive result where through the course of that amplification, you see a rise in the fluorescence channel. So in this particular schematic graph, we imagine how bright is that fluorescence on this Y axis here as a function of the number of cycles on the x-axis here. And if things go well with your assay and there's a presence of an RNA, you see this nice exponential amplification. And that's the sign that some RNA is being detected. specific, in this case, to the SARS-CoV-2 viral genome. And of course, this is the desired result here, where you do not detect the presence of the virus. Okay, so that's basically the underlying concept of how the test works. And here in just this last slide that I'll describe to you is a little bit more about how you now take that principle and you expand it and you automate it. Because as we've heard earlier in this presentation, and as I'm sure you all are very aware, a critical aspect of testing is the ability to test large numbers of people rapidly and return trustworthy results in a timely fashion. And so when we started the COVID testing lab, or the Molecular Diagnostic Lab on campus, our real goal that we put out there, and as you'll see in some of our statistics presented by my colleague Elena, we've met that goal so far, was really to keep the process sufficiently automated and streamlined as we grew in the number of tests that we were able to offer to the community, such that we could return results on a 24 to 48 hour period. And the way that we do that is by making a parallel process and using a variety of robotic instrumentation that we call liquid handling instrumentation, that essentially do the type of work that if you were to go into any molecular biology lab and talk to a graduate student or an undergraduate, you would see them moving liquids from one tube to another. And that's effectively what these machines do, but they just do it very accurately. And what I like to say is that unlike myself or our students, the robots don't get tired. So they can just keep working through the day and even through the night. And so the basic workflow here is just meant to sort of describe to you that we can take in our current pipeline, we can take samples from roughly 100 patients, and we can push them all the way through the process of consolidating those individual samples onto one so-called sample plate. We can run all of that RNA extraction work very quickly on the timescale of about 45 minutes for all 100 samples. And then those are put into this RT-qPCR test, and that takes about 55 minutes. And so when all is said and done, this entire cycle, this is a little bit dated here, it really takes about 2.5 hours, less than 3 hours, to go from raw sample to the results. And that's pretty exciting. And that's for just one of these pipelines, okay? The last thing that I just would say at this point is that there are many different types of PCR-based detection. And many of you will be familiar with the notion that at the very beginning of the pandemic, the Center for Disease Control, the CDC, developed an RT-qPCR assay and the necessary sort of design and reagents required to run this assay. And in fact, they got some bad press early on because they disseminated reagents that unfortunately turned out to be contaminated. And so they weren't very useful to the labs around the country. However, the test itself, the fundamental design, was quite valid. And so when we set out to begin this lab, we were faced with one of these decisions. We knew we could actually design a better test, being RNA biologists, but we didn't want to take the time to do it because we felt that the time was too valuable. And so what we did was we adopted the CDC test, we validated it, and I'll just point out some critical features. It has very high analytical specificity, and so what that means is that the test will not detect other types of coronaviruses, for example, or other pathogens that may be in a given patient sample. And perhaps even more importantly, the CDC assay has quite good analytical sensitivity. And so in our current experiment, we're detecting on the order of about 1,000 or 1,500 viral genomes. That's what we per milliliter. That's what we refer to as the so-called limit of detection. And lastly, the throughput, or the testing capacity per day, right now is on the order of about 200 to 400 tests per day. And as we're giving this talk, as a matter of fact, the testing is going strong by the testing, our wonderful testing technicians and team. The last point that I'll point out before handing it over to Elena is that we are actively validating a second test through interactions with collaborators and friends that we've made at Columbia University, Dr. Nisha Mishra and Dr. Ian Lipkin. These folks are world-renowned molecular virologists, and they have an FDA-approved test that is substantially more sensitive than the CDC assay. And so as we continue to pursue our goals of the Molecular Diagnostic Lab, as Jeremy will come to in a moment, that sensitivity is going to be very important. So in conclusion, for my part of the talk, as we're actively using this CDC test, we are also actively pursuing this Columbia so-called multiplex test, both because of its increased sensitivity down to just 200 viral genomes per microliter. And I'll just point out that to make this a little bit more meaningful, what that means is that in a given test, we're able to detect just 1 or 2 individual viral RNAs. That's the way the math works out. So it's very sensitive. And perhaps even more importantly, it's much higher throughput for reasons that I don't have time to go into, but that will allow us with our current staff and instrumentation to go from 200 or 400 tests up to greater than 1,000 tests per day with the same instrumentation. So with that, that's a little bit about the testing process. And now I'd like to turn it over to Elena Bosky, who will tell us more about the actual way that you go about setting up a diagnostic lab.
Thank you very much. And thank you, George, for the invitation. It's a pleasure to be here with my colleagues. So in my part, I will talk about how did we actually go about converting a research laboratory into a clinical laboratory. And it took us about a month to do this. And so first of all, what's the big deal? What's the big difference? You know, there are research laboratories and there are clinical laboratories. So the biggest difference is the purpose of the laboratory. So research laboratories, which is what we have at UC Santa Cruz. And as many people know, as most of you know, UC Santa Cruz does not have a medical center, does not have a medical school, has not been up to this point engaged in this type of diagnostic medical testing, which we are doing right now. So for research laboratories, the primary purpose is to derive new scientific knowledge, to conduct research. It's not part of a research laboratory purpose or workflow to talk to patients or to report something to the patients. And so whereas for a clinical laboratory, the primary purpose is to return results to patients so that those test results could be used in clinical care. So that's one thing. From the process side, there is a big difference in terms of the documentation and the stringency of processes that have to be in place in the laboratory. On the research side, typically researchers, would have a laboratory notebook and they will record everything they do that day. They can modify their experiments. And in fact, the whole point is that you modify the experiments until you get some interesting results that you can follow up on that would shed light onto some new knowledge. Whereas on the clinical side, there's very stringent, what we call worksheets, which are documents of what happens to every sample. There is standard operating procedures, which means locked series of steps that the technicians have to take to go from the sample to the result. There is requirement for personnel proficiency training. There is requirement to the equipment. So a lot of documentation and a lot of process changes that had to take place. Another big difference is this dealing with protected health information. So this is patient name, date of birth, so basically information about the patient, which we have to worry about because we have to make sure we report the right result to the right patient. So on the research side, we never have to worry about it because research laboratories don't directly talk to the patients. However, for a clinical laboratory, that's the integral part. And so we had to develop mechanisms to handle patient information in a secure way. So it was a lot of work from our colleagues on the IT side. And then finally, there is a big difference in oversight. So for a research laboratory, there is no license required to have a lab. There is some oversight from the university and various bodies within the universities, but there is no, state or federal oversight. Whereas for a clinical laboratory, there is this legislature that's called CLIA, or a Clinical Laboratory Improvements Amendment, which is legislature passed by Congress in 1988 that really governs the operations of clinical laboratories and establish quality standards for all testing. There is also regulation from the state. So the state of California requires all clinical labs to have a state license. The FDA is also involved because the FDA regulates medical devices, and all of these tests are considered medical devices. So a lot of oversight that we had to work out. Next slide, please. And I just wanted to point out some key regulatory relaxations that allowed us to do this quickly, even though, as I mentioned on the previous slide, this was a lot to convert a research lab to a clinical lab. And so there were some key things that happened that helps us along the way. So first of all, how did we get a CLIA license? I mentioned that a clinical lab needs to have a CLIA license. We were able to extend an existing CLIA license from our UC Santa Cruz Student Health Center that has a clinical lab on campus. And so we extended this license to a vacant research lab that we had. And this is a mechanism that is available during the pandemic. Secondly, so for a clinical lab, there is a staff licensing requirement where every staff member working in the lab has to have what's called a clinical laboratory scientist, or CLS license. And so there's been a very important executive order by our governor on March 12th, which relaxed the licensing requirement. And so we were able to employ our very talented research personnel that we have, including graduate students, technicians, senior scientists, to work in our lab. And as long as CLIA requirements could be met. So the staff licensing requirement was relaxed, but the CLIA requirements to the lab are still there. And so my role in the lab is as a California licensed clinical lab scientist to make sure that CLIA requirements in our lab are met, and to make sure that the reporting takes place to the providers, and only a clinical lab scientist or a CLS can oversee those activities. Yeah, so that's that. And then on the FDA side, so the FDA has developed what is called emergency use authorization process, which is, you can think of accelerated FDA approval process, which is not really FDA approval, but FDA approval in the context of emergency. And so they've put out guidelines for laboratories in terms of what experiments need to be done to show that the tests are high quality and could be used in the clinic. And we followed those guidelines. As Michael mentioned, we used CDC reagents. So CDC developed primers, but we made modifications to the CDC test, and probably Jeremy may talk about that later, where we replaced the limiting reagents such that we're not limited by supply chain issues. And this is really, really big insights, very, very important, that my colleagues who are experts in the RNA biology had. And Jeremy will talk about that a little bit later. Next slide, please. So I just wanted to update you on where we stand so far. So what have we done? So we started testing on May 6th. And these are data as of a couple of days ago, July 21st. So in that period of time, we have processed 1945 clinical samples from 5 different providers. Isabel have mentioned some of the providers we serve, and I just wanted to show you the breakdown. So here in purple, we have samples from our UC Santa Cruz Student Health Center, so on campus, 834. But then the vast majority actually are samples from various other providers in the community. We have in green, Santa Cruz Community Health Clinic. We have in kind of dirty green, Salud para Gente. We recently started receiving samples from the Dominican hospital. So 3 samples are shown here. And then finally 86. Those are the samples we received from Santa Cruz Public Health, so through our collaboration with the county. Next slide, please. So what type of results do we report? So we actually have 4 types of results listed here. Detected, which is a positive result, so it means that the virus was detected in this particular patient sample. Not detected, that's a negative result, meaning that the virus was not detected by our assay. And then we can also have two other types of results: an inconclusive result, which means that we cannot be sure whether the virus is there. And that's because our test is very stringent. And so when we report a positive result, we're pretty sure the virus is there. But then that comes with this caveat that we sometimes have an inconclusive test result, although not frequently. So you can see there was only 6 inconclusive results that we've had out of 1945 tests. And so in that case, what happens is we usually recommend that a clinical provider request a new sample from that patient so that we can conclusively determine whether it's a negative or a positive. And then finally, an invalid result. So that means that the specimen had poor quality, so we could not perform our test. And so again, in that case, we would request a new sample. And so you can see that most of our tests have been negative, so not detected results. We so far have had 116 positive test results as of a couple of days ago. And then we've had a couple of inconclusive and invalid results. Next slide, please. So as the previous speakers have highlighted, we really focused on having short turnaround time. That was really our goal. And so our average turnaround time right now is 26.9 hours. And I wanted to mention that this is our official, so that from the time that the sample is received at the lab to the time the official report is issued to the provider. The turnaround time for positive samples is actually more like 5 or 6 hours, because whenever we have a positive, we actually call the provider by phone, and then we alert them that there is a positive test result ahead of them receiving the official report, which will then come later once our reporting team generates that report. So this is something that we really want to maintain, and turnaround time is really crucial to our operation. Next slide, please. So this slide shows how many samples we have processed. So as I mentioned, we started in May, and we started with having 6, 8, so on average about 7 samples a day. That's what we've had in our first week. As of this past week, we're now averaging around 70 samples per day. So we've had a really big increase. And this is really a testament to my colleagues who have set up this robotic operation that Michael showed you with the robots and high throughput plate format of the assay, where we can now process around about 92 samples per run, whereas we started out with a manual process where everything was done manually. And of course, this is still very small. As Jeremy will mention next, our plan is to scale up massively from this, but this is where we are right now. Next slide, please. So the next slide shows the increase in positive test results that we have seen. So we started out with 0 positives, maybe 1 positive. When we had a positive, there was a lot of excitement, and not excitement. Of course, we don't want to see a positive, but excitement from the process point of view of, reporting this positive and calling the provider. Whereas now we really don't have a day where we don't have a positive result. And in fact, we have 10 or more. Just today we had 10. So we're seeing really an increase in positive test results, which is consistent with the data in our county. Some of you probably have seen the county dashboard that shows the case count. So we're unfortunately experiencing a surge in cases. So I think this is the end of my part. And next I will pass it on to Jeremy, who will talk about our future plans.
Thank you, Elena. Yeah, so it's good to be back again. Yeah, I think the next section here is to talk about what's on the future, what's the horizon for the UC Santa Cruz Molecular Diagnostic Lab. There's really 2 things that I'd like to point out. The first thing is we are interested and are striving to expand our testing capacity for Santa Cruz County and for our campus in general. And one thing that we learned early on in this whole process was that really the technical capabilities of the laboratory, which when Michael and I first got into this, we really thought, wow, we have these robots, we can really turn up the pace of testing by multiples of 96. And we sort of envisioned a situation where we could be testing hundreds, if not thousands of people per day. The reality of this is that there's so much more to this process than simply running the qPCR assay. The accessioning of the samples, the reporting of the results. There's a lot of steps that are rate limiting, and so a big part of this process has been trying to understand. It's not just instrumentation that needs acceleration; it's these other steps in the process. And so the entire team has been focusing on how can we increasingly automate both the accessioning and the reporting, as well as the actual handling of the samples. The other thing that has come up a couple times, I've been sort of scanning the chat, is that there's been questions about reagent shortages. And I think a common thread in the COVID pandemic in the US has been that there's a scarcity of reagents. And this leads to testing labs being unable to process samples. I think one thing that's important to point out is that very early on in this process, we made some specific decisions that allowed us to implement these tests in a way that completely avoided any of the major clinical sort of pipelines that are established. You can really see what kind of, so racket is the wrong word, but the EUA process creates a system where big companies rush to create tests. All of the instrumentation and steps and SOPs that are part of that test are essentially tied to that EUA. And then labs have to rush to get that specific instrumentation, those specific reagents. And that creates a massive demand for specific reagents. Because of our expertise at UC Santa Cruz, we focused on RNA purification and reagents that are not being used by any of the major laboratories. And that allows us to really work in a manner that's completely independent of supply chain issues. So Michael, can we have the next slide, please? So I think one of the keys to expanding the testing capacity at UC Santa Cruz, besides automating the accessioning and reporting, which is a story for another day, is increasing the amount of equipment in the laboratory that allows us to process more samples. And so currently the laboratory on the left this is our kind of operational setup. We have 2 parallel pathways, and all of our work here is focused on symptomatic testing. We're receiving samples from the clinic, and as Michael said before, we can process somewhere between 200 and 400 samples per day, depending on the need. As you can see from the data, we seldom hit that, although we certainly have the capacity to do so. As we move to this new set of primers and probes from Columbia University, the so-called Columbia multiplex assay, Michael mentioned that this is a far more sensitive test. And so this is going to enable us to shift our attention towards asymptomatic testing. So basically surveillance of individuals from the community, which should help us to identify infected individuals and provide services to them, such as quarantine, so that they can effectively be taken out of that pool of people who could transmit the virus. So using this Columbia multiplex assay, this allows us to put more samples on a plate when we run them. So that's one important improvement. The additional improvement is that because this assay is so sensitive, it opens up the opportunity to provide sort of a pooled testing strategy. And so our sort of immediate goals are expand our instrumentation, which we're going to be able to do, and also develop and validate a pooled testing strategy. And so shown below here is a little schematic to give you an idea of how this works. You can imagine in each of these groups of 8 people, these could be students or they could be people from the community. Each of these 8 provide a sample, and in the laboratory, those samples come to us. We have an initial step where we pull a small subset of each of these samples into one tube, shown here in yellow, and then each of those pooled samples gets run through our RNA purification process. In this way, we can expand our testing capacity by anywhere between 7 to 10 to even 20-fold, depending on the extent of pooling. And that pooled testing strategy is really dependent upon, or it's really influenced upon the prevalence of the virus in the community. If the prevalence is too high, pooled testing becomes a bit inefficient. And the reason for that is that once we identify a positive, as in the case in this lower right panel here, this red individual, that pool, based on the process that we're using, can be disaggregated, and we can retest the individual samples that make up the pool. And so that allows us to identify that person. And in the case of this pool testing scenario, we talked about how our standard process is maybe between 2.5 to 3 hours. In the case of the pool testing, we'd still have that 3-hour kind of time frame, but once we've disaggregated those samples, they would be retest in our sort of standard pipeline as an individual sample. And so that would add probably an additional 3 hours to the time that it would take us to identify an individual who was positive using a pooled testing strategy. So we see this as being a very powerful tool that we can add to our arsenal. And there's a number of ways that we could apply this. And so if we can go to the next slide. One of the main goals, I think, and the hot topics in certainly higher education is how does higher education or institutions of higher education return to normal campus activities in the pandemic or post-pandemic era. And I think, you know, we all recognize what an incredible place UC Santa Cruz is. It's a beautiful place. But as our chancellor says, you know, the university is really, it's the people that make a university great, not necessarily how beautiful it is. And so right now we're missing a huge fraction of the people that make our campus so fantastic. And so we really want to be able to get back to the point where we could bring students back to campus, bring our full research labs up to their normal capacities, and do so in a safe way. And so we are envisioning applying this pooled testing strategy to our campus in a way that will enable us to test most of the campus repeatedly over the course of many weeks. And this will allow us to, again, identify cases of asymptomatic transmission that could then be hopefully pulled out of that pool and allow us to break transmission cycles before they really begin. And this is work that's being done in collaboration with a massive crew of different departments on campus, as well as our colleague Martin Kilpatrick, who's helped us with the modeling. Okay, so can we go to the next slide? And so, you know, in order to really make this happen, there's a few things that need to occur. The first is that, I guess this is like a no rest for the weary kind of thing, but we have to establish our regulatory roadmap. So this falls on Elena and Isabel to help steer the lab through the validation process for our pooled testing and for our multiplex assay. Our technicians are in the process of validating this pooled testing strategy. And more importantly, we recognize, I mean, I've been swabbed many times now, I think 8 or 9 times, that a nasopharyngeal swab is not going to be something that we can do in a high throughput manner. So Michael mentioned there are other biospecimen types that are out there, so saliva and anterior nasal. And so we are really interested in validating these different sample types in our process, which will enable either at-home collection or supervised self-collection. So it'll take the healthcare provider a little bit out of that process and enable them to conserve some PPE and things like that. But we imagine it'll also make the process of giving a specimen much more palatable to people in the community. And finally, we have a lot of new instrumentation coming into the lab. All of that equipment needs to be validated. And then the last big step is to develop information management systems, which are currently nearly finished, that will enable us to have patient ID associated with barcodes on tubes that come into the lab and are instantly accessioned, and then the reporting can be done electronically. So we think that this is going to be really the key to upping our testing capacity. So there's, I guess, lots of more work to do, and the team is going to be super busy over the next 6 weeks, both with our ongoing testing and developing all of these new approaches. But it's something that we're really excited about, and we think we're going to be able to have a positive impact. So with that, yeah, just thank you very much. It's been great, and we look forward to discussing maybe some funny tales from our experience.
Thanks, Jeremy and Michael and Elena. This is the point at which we want to go through questions that all of you have sent us, and you're still welcome to send it as we're talking. We'll try to get to as many as we can. There were several questions around the categories of partners that we have, and whether or not we are testing just students on campus, whether we're also testing staff, why Dominican numbers are so low. So I thought it might be helpful just to give a really quick overview of that history. We started May 1st, so very recently. When we started, obviously, as you heard from this presentation or from Elena, this was very new. There is no hospital on our campus. We created this lab and learned what all the diagnostic requirements were and all the regulatory requirements. So we started small, very purposely. We have an extraordinary partnership with our Student Health Center. We wouldn't be able to exist without our Student Health Center, and not only because we work off of their license, but also because they have worked with us through this whole process to provide good diagnostic testing and reporting. So through that health center, we test students. We also test employees that are working in positions that are at risk. And so that's the answer to the staff and the faculty. Community Health, Santa Cruz Community Health was our first community partner. I mentioned that. Salud para la Gente is the next one in line. We had an early pilot with the county, and we are now working with the county to roll out emergency testing where it is needed with several partners. So the Dominican results that you saw are through those emergency agreements. At the same time, we are working with local hospitals, we're working with the county jail and other potential partners to start testing on a higher level. So that's the answer to why you see the distribution of the numbers that you do. So one of the interesting things about what we're doing is that it is almost 24 hours, and there's an incredibly dedicated crew that has been working around the clock to do this testing. And one of the things that is very real about it is that when you get a positive result, you need to call and let the provider know immediately. And so, in the state. So Elena is leaving our talk early because she needs to make some phone calls about the positives that came into our lab today. So I would like to direct the first two questions to Elena and then move on to questions that are particular to Jeremy and Michael's expertise. So Elena, there are some questions around reporting. There was a question about whether you report to CalReady and how the reporting process works. And perhaps you could give an overview of that process.
Absolutely, thank you for that question. So we report to 3 different places. So we report to the provider, we report to the state via CalReady, and then we also report to Santa Cruz County Public Health. The reporting is still, so as Isabel mentioned, we started from scratch, and so we had to figure out the systems that hospital has and they're native to them in terms of reporting and electronically connecting to medical records. We don't have that, and so we started with manual reporting, which means we have a technician who creates a PDF and sends via secure fax that PDF to the provider and to the county. And so that's definitely the bottleneck. So let me tell you about our process, because I saw that there was another question about the timing. So samples typically come into the lab around noon or so 12 p.m. Then the results are available around 5 p.m. And then the calling of positives happen after that, so around 6 p.m. or if there is a delay, 7 p.m. So that evening we call the providers right away. But then the actual report, because I mentioned this is a manual process right now where we have to make the PDF report or prepare another format that the provider wants. So that happens the following morning. The reporting teams comes in and sends out all those reports to the various places that I mentioned. So that's why there is a delay. But we're moving very actively and very aggressively to electronic reporting, which is a big undertaking for us because we didn't have that in place at all. And so we had to learn how to do this and get expertise and work with other providers on that.
Thank you. There's also a question, Elena, about whether we have an EUA and what the associated process is around that. Perhaps you could give a quick overview of that process.
Yeah, so FDA has put out guidelines for how to get an emergency use authorization for your test. And there is two types of guidelines. There is guidelines for manufacturers, so people who want to distribute their test. They could go through the EUA process for manufacturers. There is a separate EUA process for laboratory developed tests. I saw somebody asked about that. Our test has been validated as a laboratory developed test, which means it can be run by our lab, so it's specific to our lab. And we have submitted an emergency use authorization request for our test and have been in communication with the FDA. In fact, FDA just asked us to do some more experiments, which we have now done. And so we will be resubmitting our EUA very shortly for the first assay that we talked about. And then for the Columbia assay, that will be also a separate LDT that we will be validating and applying for emergency use authorization of that LDT, a laboratory developed test.
Thank you, Elena. I understand you need to go off and report the positives, so thank you very much for your participation here.
Thank you very much.
So there are a number of questions around false negatives, and I wanted to direct this question to Michael. There are a number of questions that essentially ask, can you describe what the false negative rate is? How does that happen? What is it about a test that makes a false negative or does not? And associated questions around, will that change in the lab over time or not, based on what the future plans are?
Yeah, thank you, Isabel. That's a really critical question. The issue of false negatives and false positives both come with a number of very really important implications. So on the false negative side, I would say, at our stage, we've done quite a lot of validation work, and we've used that word validation a lot in the lecture today. A lot of that is done with what is so-called human contrived samples. So in the early stages of setting up this lab, and we are essentially going through the same motions that many of the testing labs are doing, there weren't enough available positive samples to do really rigorous statistical analysis on false negative rates, false positive rates. So we follow FDA guidelines. We use synthetically generated RNA molecules that are the same sequence of the SARS-CoV-2 virus to run our validation. So, the question of what is our false negative rate, the simple answer to that is that we have not done enough testing to really reliably know the answer. What we can say is that we tend to use the term limit of detection, and also built into our result reporting structure, as Elena mentioned, when we report a result, we actually don't use the term negative when we report to a provider. We use the term not detected, and that's because any assay, as we've discussed, or any test rather, that we've discussed, has some limit, some threshold limit of detection. And so when you're not detecting it with a given test, you cannot be 100% certain that there's no virus in that sample. And of course, as you improve that limit of detection, or improve that sensitivity, bringing it lower and lower, which is what the multiplex Columbia assay is going to provide for us, then that whatever that false negative rate may be at present will be suppressed even further. And so that's some of the thinking that goes into, how can we be sure, or why do we choose particular language with reporting as detected versus not detected, as opposed to the word negative, because there's always some threshold limit of detection. And so I did notice, related to this question, is this notion of, have we compared to other laboratories our results and these sorts of things. And I'll just mention that in that validation process, there's a lot of comparator assays that are going on. So we do, in fact, have to send our samples out with our calls of detected or not detected to other FDA approved labs to make sure there's correlation and agreement. And we also actually receive samples from other labs. And so the laboratories interact quite a bit to get exactly at this, this question of how accurate are the results that we're reporting. And thus far, I'm proud to say that our results are lining up very, very well compared to colleagues at other testing laboratories.
Thanks, Michael. Building off of the question and Michael's answer, Jeremy, we have a specific related question around asymptomatic testing. So do asymptomatic patients typically have a lower viral load, requiring greater sensitivity in the testing process?
Yeah, that's there's certainly literature on that, and the higher sensitivity of this Columbia test that we're going to be running is going to be critical for identifying patients that are people who have much lower viral loads, and that is the general trend with the asymptomatic individuals. So it's really, and for validating this test, and this is an important part, the FDA has specific guidelines for validating asymptomatic testing, and so in that process, we have to be able to screen a much higher number of clinical samples that come from other laboratories that are known to be negative, known to be positive, and show that our test correctly identifies these individuals. So that gets at that question of false negatives. Yeah, so that's a key part of identifying these asymptomatic individuals.
Thank you. Okay, so this one is going to be interesting because I think you two will end up tag teaming it. We have a lot of questions about pooled testing, a lot. And if I break down what those questions are, they are essentially around, can you describe it a little bit more? Can we understand better how it works? And equally, what happens with pooled testing if there is a positive in the pool? And then the third piece of that is, what kind of population fits pool testing and what kind of population does not fit going down the pool testing route? So if the two of you can figure out how to balance that one, that would be great.
I mean, I'm happy to start. I think from our process standpoint, the pooled testing strategy is really this. Let's see. So we got to start with the process. So yeah. So the idea here is that we're going to be from samples that we've collected from individuals through our normal approach, nasopharyngeal swab or oral swab or anterior nasal swab. Those samples are their own individual collection tubes, and we will be pooling, grabbing sample from those tubes and combining them into a single tube. And that single tube will be processed through our RNA purification pipeline. And so that way, with respect to our 96 well plates of RNA of samples that would get processed for testing, each one of those individual wells will contain multiple individual people. So currently, we will be looking at pooling different numbers of samples in our validation experiments. The important distinction here is that the sensitivity of the assay is obviously proportional to the number of individuals that you've pooled together. And so so that means that we're going to be essentially diluting out the positive viral RNA in these samples. So when we do this pooling, the really exciting bit about how we're planning to do this is that all of our tubes that we send out for our sample collection kits are barcoded. And so we understand patient identification is associated with those barcodes. And so we know which tubes went into which well of this plate. So that means each individual can be tracked. We know what rack that came from because the racks are barcoded. And once we get a positive pool result, we can go back to the racks that contributed to that pool and then grab those individual samples, those 10 individual tubes, and then retest them individually in order to identify the positive sample. And so that result can then be directly reported via the processes that Elena talked about.
And I'll just pick up on that a little bit. So Jeremy really just outlined the sort of underpinnings of the pooling process, but another part of the question that Isabel framed was, when is pooling appropriate? What is a population that makes sense to pool, and why would you pool? And so there's a couple things to think about there. One thing to think about is pooling works best in theory when the prevalence of the virus is quite low. So in other words, if you have to do thousands of tests to find a single positive, then you're better off pooling and having to do hundreds of tests to find that same positive. So that's one thing to think about. When the positivity rate or the prevalence gets too high, at that point, it really doesn't make sense to pool. So that's one thing to think about. And you can play with the math a little bit and model that, and many people are doing that and publishing on those subjects. And we'll be, as we develop our own pooling strategy, we'll be working very closely with Marm Kilpatrick, who is an expert in this area and is really studying all the models. The other thing to think about when you ask the question, when does it make sense to pool, really comes down to a matter of the reality is that even without supply chain interruptions that Jeremy mentioned, so we're not suffering from that, thankfully, due to some strategic decisions made early. The reality is that everyone has some limit to the number of tests that you can run, both in time and just in terms of staffing and in terms of actually on the front end clinical provider, the ability to see all the patients. That's why the idea of supervised self-swabbing is very attractive to us to expedite that. But another key feature that I would just raise in the answer to this question is that, and this is a really important point about testing generally, so it applies to pooling, but it's also more general, is that when you're tested and you receive a not detected, or often they might say negative, but virus is not detected, what you're really getting is a point in time. So you're basically getting a moment in time when you are not testing positive for the virus. In instances where we really want to control the spread of this pandemic, which of course we do, one must continuously test individuals. So we refer to testing cadence as sort of a term of sort of how frequent must you test the population so as to pick up people in the infection cycle with a high sort of penetrance, let's say, like you pick them up often, so that you can then isolate them, right? Remove them and break that transmission cycle. So when you get to issues of numbers of tests, and then the very important concept of testing cadences, and there was a very important paper that was published, I guess, just under a month ago or so, maybe a little more recently, that suggests that individual populations, right, should be tested on the order of every 3 to 5 days continuously. And so in order to capture those people who have the virus early on enough in the infection cycle that you remove them, you quarantine, you isolate, and they don't spread. And so pooling is, as I'm sure many people on the call will know, was just recently FDA approved for the first time just last week. And there's a lot of attention being paid now about pooling. I'm not surprised that there's a lot of questions. And this is the reason why, is that in a world of limited testing, even if you have enough supply, to test at a population level with cadences that are effective to control the and break the transmission cycle, pooling, I think, is going to play a very important and critical role.
The other piece to that is that the cost per test per individual drops, which is another nice point. I've seen in the questions, there's been lots of comments and concerns over what it costs to actually run one of these tests. And while it certainly isn't what the big clinical diagnostic labs are getting paid, it's not, for an academic institution like ours, it's not necessarily cheap either. And so I think when you get to that ability to pool those 10 or 20 samples together, you have a cost reduction.
That's actually a very good segue to one of the questions we have here around funding, actually. So how are you funding all this work now and into the near future? How are you, if at all, coordinating with broader UC system-wide testing scale-up efforts? I'll start an answer to that, and then I think you guys will come into this as well. The campus has been absolutely incredible in terms of stepping up and funding a lot of this work. As Jeremy noted, this work is expensive. We are trying to think of ways to economize all the time and be efficient and effective in what we are doing. There is no question that we rely on philanthropy to help us in terms of funding. The Community Foundation has stepped up and is contributing support for testing the safety net clinics, which we describe as, at the moment, there are several. So there's Salud para Gente that we talked about. There's Santa Cruz Community Health. In addition, they've stepped up to help with funding when there are emergencies, right, where there's real risk. And so is the county. And so that's how the funding is happening. It's very grassroots, and assistance is absolutely needed. There's no question about that.
And then I guess there was a comment on, are we coordinating with the other
Right.
UCs? And I'll just bounce that over to Jeremy because he was part of an important UC-wide committee. So why don't you?
Yeah, there's been a task force that's been meeting regularly since the lockdown began, that brings together all of the big players in the medical diagnostic community that are at the UC medical centers, as well as people in the public health realm and the legal aspect of public health. And this task force was really focused on modeling the transmission of the virus on campuses in the UC system, also focused on innovative testing approaches, as well as testing capacity on each campus, and then finally on the contact tracing side of things, and also the trying to understand what services are going to need to be provided to UC campuses as the students return to the classroom or return to campus. And so it was a really interesting group, because certainly in the diagnostics working group that I was a part of, I would say early on, there was a lot of sort of skepticism that research campuses like ours or Berkeley could roll out and set up a medical or a clinical diagnostics laboratory. And there was, I mean, I would say outright shock that this could happen. We were often told we were practicing medicine without a license and no malpractice insurance. And there was a lot of real sort of perspectives that, you know, this is not something we should stay in our lane and stay in our lane. And so as this went on, and you began to realize that these big CLIA labs that are running all this diagnostic testing for the hospital systems, they're not a very good source for innovation, and they're not really all that well equipped for dealing with a pandemic where you need massive amounts of testing, because they're constrained by supply chain, they're constrained by their approaches, and they also have a big burden of their actual hospital systems that they're affiliated with, all the tests that they need to run. I'm sure many of these laboratories would prefer not to be running COVID tests. That said, it's amazing what the medical campuses have done for California. They are testing at a huge level. And when you see the commitment that these laboratories have, it's absolutely amazing, and how they've repurposed their infrastructure to get this done. So there's no doubt that the University of California medical system has had a huge impact on testing in California. The things that were really interesting are some of the other incredibly innovative approaches that have been coming out of UC research labs. At UCLA, for example, a technique has been developed using next generation sequencing that has the potential to scale incredibly. Essentially, it's still a nucleic acid test. It has all the same aspects of our test that we've been talking about, but the number of samples that can be processed simultaneously is much, much larger. So you're talking on the order of tens of thousands of tests in a single shot. And there's been a few other innovations out of the UC system that could have a big impact, if not on this pandemic, on the next one. So that task force, it was interesting because it really only provided guidelines. And here are different tactics that UCs could use. And what they recognized was that each of these campuses are very distinct, and they have different needs and different interactions with their community. And so it's been a document that's guided how our campus has developed its reopening plan and its testing program and the services that it plans to provide. And I think we've put together a really robust and rigorous approach for helping to bring the students back, that'll keep them safe and keep the faculty and staff safe. So I think it was there's definitely a lot of interaction between campuses.
I'm realizing that we're getting close to the end of our time, and we have some interesting intersecting questions from some people who clearly know what they're talking about. So we have some questions about our LIMS. And LIMS, for everybody else, is a laboratory information management system, which is essentially a data management system that makes ramping up testing possible. And then the intersection is that there are a lot of questions around what kind of specific challenges did we face as a lab. And so, Michael, I was hoping that you could speak to what the LIMS serves for us and how it's addressing many of the challenges that we're facing.
Sure. Right. So I'm going to go on the record as saying, as a biochemist by training, that prior to this experience, I did not know what LIMS was. And yet now I know very well what it is, because we, together with a custom software company, have essentially built from the ground up a custom LIMS system. There are essentially three critical features of a laboratory information management system, at least of the type that we have built out. There's a front end that we refer to as the portal. There's a sort of a middle section, which we refer to as the core LIMS, and then there's the back end, which you could think of as the reporting structure. So the portal really relates to this intersection of how is LIMS helping us and what are the big challenges that we face. And so one of the really eye-opening observations that we had as a group when we first started out on this adventure was when we began testing, as Elena mentioned earlier, the reports were physical documents, PDFs that were then actually printed out, walked over to our UCSC student health clinic, and if you can believe it, faxed to the clinical providers. And that was pretty surprising, I guess, to me, and yet it's not terribly uncommon in the medical communities to be using robust, secure, yet one might argue antiquated technologies. And along with that was sort of a desire for many clinical providers to use, and this still exists to this day, and these are ongoing negotiations, to use paper requisition forms. So for every individual patient, there's a piece of paper with all the PHI and protected health information. And so that front end portal is basically a way to create electronic or digital requisitions. And so that is something that is really just coming online over the last couple of weeks. And together with these barcoded tubes that Jeremy mentioned earlier, that dramatically streamlines not only the processing of the samples, but the way that the information moves around in a very secure HIPAA compliant fashion, so that when requisitions are generated at the provider and then samples arrive in the laboratory, we can take these samples, we can put them into these neat little plastic racks, and 96 tubes at a time, we can put them on a little glass plate scanner, and in 3 seconds, scan the ID of all of those 96 individual samples, and then that matches into our LIMS system to the requisitions that were created all over the county at the different medical providers. And so that's a huge, as you can imagine, that's a huge boost in sort of the information flow, which can often be a limiting step in the testing process. I've seen several questions about this. If you're saying you can do 200 to 400 tests a day, why are you only testing, say 75, or now we're sort of tending to average around 100? And there's a lot of answers to that. But one part of it is that, as was mentioned earlier, there's a lot more than just running the test that goes into safely and accurately testing patients. And so the LIMS is going to remove many of those pinch points or bottlenecks in our process. On the front end, on the interior sort of core LIMS part, and that really relates to what we inside the lab, as samples are moving from each step, you could think of it from each robot and point in the process that I described, that's all being tracked through barcode scanning. And so the sample status is just sort of moving through. And so right now we're filling out traditional CLIA compliant paper worksheets, and we're saving all of those as we're required to do to be CLIA compliant. This will all be now made electronic. And again, that will essentially allow us to do more testing, maintaining. I always talk about the idea of, it's great to scale up capacity, but not if the integrity of the test is compromised. So we are uncompromising with respect to the integrity of the result of the test. And then the last bit of the LIMS, which is maybe the most important part, is the reporting structure. And so we heard a little bit of this from Elena, that you're obligated by state and federal law to report these results, particularly the positive results, in a timely fashion for obvious reasons, so that they can be clinically acted upon. And there are several entities, both local and at the state level, that need to receive that information. And so what that reporting component or module of our LIMS system is going to do is essentially send out the signal straight away. There still will be a human review process, which is very important, just to make sure that no mistakes or swaps are made. But then with a touch of a button, all the sort of necessary agencies that need to receive that information will get it essentially instantaneously. And so that's quite a lot about the LIMS. And then the general question of what are the major challenges. I see that we're running low on time, so I don't want to. There are many, I will say, but I'll flip the question around to just conclude, which is that we have met many challenges, but we have not met a challenge that we couldn't overcome yet. And that is a testament, not to the people who are on this call, unfortunately, but to the people who are not on this call, who are still at the laboratory as we speak, and show up very early and stay very late. And these are graduate students, research staff, and people who never could have imagined that they'd be applying their scientific skills in this way, and have just risen to the occasion. And so I just wanted to give that credit where credit is due at this sort of concluding point.
I think the only challenge, like some of the challenges that are more concrete that I think we faced and still do, is definitely being, and this is common to other pop-up labs, is actually being able to get samples into the lab from different healthcare providers. There aren't good mechanisms for a non-medical campus like ours to begin to bring in patient samples and to recoup funding from insurance agencies and to do billing and everything else that a typical molecular diagnostics lab would accomplish. And so I think it's taken our community a little bit of time to adapt to how our process works. But what we're seeing now, especially given that we have capacity, is that people, it's kind of like if you build it, they will come. And that's certainly what's happening. And I think probably the most interesting challenges has just been within this amazing team, certainly the healthy tension that has been has developed in our group in terms of taking this problem and this purpose very seriously, but at the same time, wanting to push as hard as we can in the direction of expanding our tests, but still maintaining the quality, like Michael said, balancing that with reporting and really being able to always see eye to eye. I think sometimes during this process, one or two of us have pushed too hard in one direction or another, and it's been interesting to see how those dynamics have played out. But in general, we have this just this amazing team that has really, I think, risen to the occasion. So I think the challenge is just standing up a lab that has never been stood up on this campus before, and it's been pretty fun to work on this.
Thank you. I'm conscious that we're running out of time or have run out of time. And I just want to say, I want to echo Michael and Jeremy, like people have been amazing. It's taken really, truly a village to make this happen with this lab. And from the county to our own students, to our own faculty, to our campus staff and leadership, it's really been incredible. And so we're very grateful for that. And I think it's a testament not only to the university and this team, but also to the fact that we're really watching people come together with COVID-19, and there are incredible opportunities that are coming from those collaborations that I hope live long after this pandemic is solved for all of us. So thank you. Specific thank you to Rafe and to George for sponsoring this event, for making these events happen, because it's a great opportunity for us to be able to tell everybody what we're doing and to hear from you about what you're interested in. So thank you.